Alright, so this class is Biochemistry 1, so make sure you guys are in the right class. This course will be taught in English, so that will be my job to make you guys understand at least a little bit about biochemistry. I mean, not supposed to be a little bit, but it's just that. We hope that you will get some stuff out of this class, okay? So today is actually going to be a semi-review of what you should have learned in chemistry, general chemistry. Also, you will probably see a lot of the information that you learned in high school. Okay, so today is not very serious for me to go over the syllabus, go over the scope of biochemistry, the things that we will cover. So first thing first, course syllabus. Okay, so this is the course syllabus. So I'm going to go over the 18 weeks of stuff. So the first third of the class will be focused on very general stuff, things like scope of biochemistry. Primarily here, we're also going to be talking a lot about instruments that we use in biochemistry. Okay, so we'll also talk about that. And of course, throughout our discussion this semester, we're going to sort of introduce to you guys a lot of different instruments that we use in biochem. Okay, so that next semester, for those of you that are in our department, when you take the biochem lab, you'll be like, huh, okay, so I remember a little bit about that. Right? So, we'll talk about scope of biochemistry, very simple stuff. And we'll also talk a little bit about the energetics of life. Okay, so energetics here, we're talking about things related to energy. Okay, so we are living, so of course we need to get energy from something. Right, so we're going to talk a little bit about the chemistry aspect of that. And then for 4th to 6th week, we're going to be talking about nucleic acids. Okay, so we're going to go over the characteristics, the structure, the function of nucleic acids. Okay, and obviously nucleic acids are things like DNA, RNA, and that kind of stuff. Okay, so that's that. All right, so the first third of this class is extremely boring. Right? It's not very exciting. It's just general stuff, but things that we think you should know. Then we get into the second half or the second third of our class, and here we have proteins. Okay, so the second third, we'll primarily be talking about proteins. Okay, so proteins are important for a lot of different things. Okay, for example, enzymes to catalyze chemical reactions, for functions such as supporting structures. Okay, so we're going to give a very general introduction to proteins, which also includes things like amino acids, amino acid structures, and characteristics, things like that. Okay, and then we're going to talk a little bit about the three-dimensional structure of proteins. Okay, so proteins are very awesome molecules that can do a lot of crazy things. Okay, so we're going to talk about that, like why do they do it, why can they do it, things like that. So continue to talk about 3D structures here, and then we're going to talk a little bit about protein functions. Okay, protein functions and evolution. And then if we have time, we'll talk about contractile proteins and molecular motors. This is somewhat less important in terms of our discussion, because you'll hear this again when you take cell biology. Okay, cell biology, you will hear this over and over. So that should be plenty of time over there. All right. So the third of the course, this is actually much more interesting. Okay, so I can talk more about these things. So we're going to try to get to these things earlier if possible. Okay, we're then going to talk about carbohydrates. Okay, so you guys know carbohydrates for the the people that really care about their diets in the audience hear about carbs as something very evil, but in reality, carbohydrates are really good stuff, okay? They not only account for energy that you get, they also account for structure, okay? So we're going to take a look at some examples today. Right. Also, then we have lipids on week 14, okay? So chapter 10. Lipids, we'll talk about what lipids are, sort of what are they like, what are their characteristics, what are their functions, that kind of stuff. We'll go over lipids. And then week 16, we'll start talking about enzymes, okay? So previously, on the last slide, we talked about proteins, like general proteins, okay? But then when we get to chapter 11, which is a very important chapter, it deals with enzymes, okay? So enzymes are the core of biochemistry, okay? So nothing else, enzymes you definitely should understand, okay? Because, you're sitting here breathing and alive because of enzymes, okay? So you got enzymes in your body catalyzing thousands of reactions at the same time, and we're going to take a look at what are some properties of enzymes, okay? And then chapter 12 is a chapter called the chemical logics of metabolism, okay? And we're going to talk a little bit about that because metabolism is biochemistry too, okay? So today, or this class is Biochem 1, okay? So Biochem 2, it's all about metabolism, okay? So chapter 12 is sort of like a preview of what you need to know for metabolism. Biochemistry is not difficult. To my opinion, it's a lot easier than things like calculus, 像微积分之类的东西. It's seriously a lot easier. It's a different type of science. Right? You have to understand the chemistry behind it in order to understand all this descriptive science. Right? So biochemistry is a very interesting topic. Okay, so this is what this semester will look like. So before we get to the serious stuff, just a little introduction about myself. I started working here 3 years ago. All right, so you guys are the third biochem class that I'm teaching. So prior to that, when I was in college, I wasn't studying in biology per se. Okay, I was a chemistry major. Okay, so what you're gonna hear from me in terms of biochemistry is probably more on the chemistry aspect. Okay, and the reason for that is because this thing is called biochemistry. Okay, so biological chemistry. Biology is the adjective, chemistry is the noun. Okay, so this is a chemistry class. Okay, so it's not a biology class. Okay, so if you want to take a biology class, I don't know if you have this, but there's this thing called the chemical biology. Okay, so different things. Right, different things. Right. So here we are really studying chemistry. Right, so it is a chemistry class, but related to biology. Okay. Let's be clear on that. For the majority of you guys are a second year undergraduate student in the Department of Biological Sciences and Technology, biotech. Right? So you guys are the biotech majors. For you guys, this thing is sort of the fundamental core of everything else that you want to study. Okay, so this is why we decided that it's a good idea to put it in the second year. Right? Actually, for me, it's actually a bad idea. Right? Why? Because some of our professors think that you should have organic chemistry before you take biochemistry. But the current design is that you take organic chemistry 2 and biochemistry 1 together. Okay? There is a benefit. The benefit is that when you are taking organic chemistry 2 and taking biochem 1, you kind of sort of see the link between biochemistry and organic chemistry. That's the benefit. I think that's the only benefit. Right? The downside is that you're taking 2 really serious chemistry classes at once. Okay? That puts a lot of workload on you. Okay? So biochemistry is the branch of science that seeks to describe the structure, organization, and function of living matter. Okay? So we're going to talk a little bit about the definition of living matter a little bit today. All right? So really, we're going to be looking at structure, organization, and function. Okay? Oh yeah, by the way, the textbook for this class, it is still the same textbook as previous years. The title is called Biochemistry. The author is Matthews something, 4th edition. It's the same as last year, okay? However, sometimes in my slide, you will see pictures that do not belong in the textbook. That is because I also use another textbook called the Leninger's Principle of Biochemistry. Okay, so it's another textbook. Right? It's really just to help you, right? Because you have your textbook. So if I just, copy and paste the textbook's picture onto a slide, it's kind of useless. Sometimes 你要是发现说，那个图片是从哪里来的？不在我课本里啊。That's because 真的不在你课本里。Okay? Really, the idea is for you to listen to me talking and learn from the slides. Right? The book is always just a reference, but there's a lot of information in the book that we cannot possibly cover in the semester, okay? Or actually in 2 semesters even. Just 课本里面有太多 information, 我们没有办法全部 cover. 所以我们一定会挑 the important stuff, okay? So we'll select what's important, what I think is necessary to get you to, your future career or your graduate school if you guys are interested in graduate school, right? So I think the stuff are important, so they're on the slides. All right, so the goal of biochemistry is to understand life at the molecular level. Okay, so really, we're talking about the chemistry of life, okay? Life is an awkward word. Before you got excited, we're not talking about people and people, okay? We're not talking about life in a philosophical way. No. I would actually be more excited about a class like that, but unfortunately, we're dealing with things like this, all right? Yeah, so this guy, it's a cell. I hope that's obvious, all right? So you go from a cell, okay? We know cell is like a little basic unit in your body. And then take the cell apart, you got things like organelles. Inside the organelles, you have different things. For example, inside the nucleus, you got chromosome. Chromosome is made out of DNA. DNA is made out of little DNAs, okay? Nucleotides. You got the membranes that look like that. Membranes are made out of proteins and of course also lipids. And then lipids are made out of fatty acids. Proteins are made out of amino acids, things like that. Okay, so the stuff that we're studying is really the fundamental components of life. Okay, what makes life life? Also, really the field of biochemistry is very important because it sort of links the genes to phenotype, genotype to phenotype. Okay, so the stuff that we look at are the phenotype. For example, why you have yellow ducks and black ducks, okay, because they have different pigments inside of their hair. Okay, so things like that we will talk about. Biochemistry primarily can be divided into three principal areas. Okay, the first principal area is called the structural chemistry. The structural part is the structural chemistry of the components of living matter and the relationship of biological functions to chemical structure. Okay, so really we're looking at this. Biological function to their chemical structure. Okay, so this is a chemistry class again, so we're going to be looking at the chemical structures. Okay, hopefully you guys are comfortable with chemical structures. Okay, and we're going to be looking at chemical structures and then correlate that back to their biological functions. Number 2, we have metabolism, and metabolism is really the totality of chemical reactions that occur in living matter. Okay, that means that in you right now, just by sitting here, there are probably thousands of chemical reactions happening to you or in you, right? And together we call that metabolism. Okay, so all of that collection of things, chemical reactions that happen in your body, we call that metabolism. Okay, and that is biochemistry too. And then there's the third part. That's called the genetic biochemistry. Okay. And genetic biochemistry is the chemistry of processes and substances that store and transmit biological information. Okay? And we will not talk about this in biochemistry too much. We'll talk about the structure of DNA, the actual mechanism of DNA replication, DNA blah, blah, blah stuff. We leave that to molecular genetics. Okay? So for those of you that are in our department, you have 2 semesters of molecular genetics. Okay, so that we leave it for those guys to teach. The third area is also the province of molecular genetics. Right, so we will only cover these 2 in biochem. And we will only cover number 1, this semester. Of course, we start with the 4 biomolecules that we have in nature. Okay? So this class primarily discusses in detail the properties and functions of these 4 major biomacromolecules. And the 4 classes are carbohydrates, proteins, lipids, and nucleic acids. Carbohydrates primarily is used for energy storage. It's used for structural support. Proteins is used as enzymes. That's a major function of proteins, but also structural supports. You got lipids, fats. Fats is the same thing as energy storage. And it's also used as cellular membranes. Okay? And what I'm also missing here is a lot of time cell signals. For example, cholesterol, hormones, that kind of stuff. Okay? 有没有知道 cholesterol 是什么？我看过一个广告，胆固醇的广告，就有一个很胖的胖子，然后趴在另外一个人身上。有看过那广告吗？对不对？胆固醇嘛，对，胆固醇是一种 lipid， right？ 那还有什么东西其实也是 lipid？ 很多荷尔蒙类型的东西也都是 lipid。你就会发现哦，荷尔蒙其实它是 synthesized from 胆固醇。OK， 所以下学期会跟各位介绍怎么样子去做人体的不同的荷尔蒙。OK， 那荷尔蒙有很多 function 啊，例如it triggers development or what it can also do is it can suppress immunoresponses, things like that. It's very diverse, right? So lipids has a lot of other functions as well beyond just fat. Nucleic acids, DNA and RNA. DNA and RNA, they're primarily used as information relays, but also RNA can do a little bit of catalysis. Carbohydrates, we have some examples like bread and sugar, okay? So things that you eat, but also they make up your plants, okay? So the reason why plants could just stand there and, survive a hurricane is because they have these strong carbohydrates, okay? Also, crabs, okay? Crab shells, insect shells, these shell things are also made out of carbohydrates, okay? So carbohydrates is very interesting in a way that depending on how it's organized, you can have very different properties in terms of strength. Okay, so carbs are very, very interesting. Proteins consist of enzymes, structural supports, etc., and other diverse functions. For example, proteins can help your bacteria move around, like this little thingies. It sort of spins, and then your E. coli can fly around. Your red blood cells, okay? Inside of these, you got a lot of these things called hemoglobin. Hemoglobin does what? It carries molecular oxygen. Okay, so it binds oxygen. So it has that function. Eggs, it has a lot of proteins, right? It helps things to grow, right? So proteins. Cows, milk, okay? Nutrients. So how you can pass nutrients from an adult to a child, things like that. Muscles, it supports your physical strength. Okay, so those are also proteins, right? So proteins do a lot of stuff, right? So that's really, remember, enzymes and proteins are the core of biochemistry. And then lastly, you got this ugly figure. Unfortunately, that's how we visualize proteins. Proteins are made out of amino acids, and they form different structures, okay, like the secondary, tertiary, quaternary structures. We'll talk about that. There's no better way to visualize it on a computer than that, okay? So if you guys in the future for your career wants to do, I don't know, drug design, you have to know this, okay? Lipids account for fat, cellular membranes, and hormones, okay? So a lot of times lipid can store tons of energy, okay? They're used as a long-term energy storage. Soaps, okay? Soaps are fat. So if you go and take a bunch of animal fat and you pour NaOH in it, sodium hydroxide, you make soap. Okay, that's actually how you make soap, right? Cell membranes. Wow, isn't that interesting? So you got like lipids all over. These are all lipids, okay? Those are all lipids. So they make your cell membrane. Nucleic acids, we have DNA, RNA, and yeah, nucleic acids are really boring. There's no better way to put it. They transfer information, okay? So they do a lot of information storage. There's a increasingly interesting field of research, okay, that look at, is it possible we use DNA to store information for a computer? So that's like a new research field that people are really interested. Why do we want to do that? because for example, one tiny bit amount of DNA, you can store a lot of information inside, okay? Have people made progress? I don't know. I just know they're doing it, but I don't know if they made significant progress yet. But just so you know, if you want to do that. Molecularly, this is what they look like. You got different bases, okay? They base pair with each other. That's how DNA can multiply, all right? And obviously, DNA is also related to genetics. Have you guys seen this advertisement? You have, right? And have you also heard about this female actually sued the company because she didn't do any plastic surgery? Because she was a model and she posed it for this clinic, and then this picture just got out and everybody's like laughing at it. But yeah, genetics, information transfer between parent to children. Of course, nowadays, it's very difficult to tell, right? Like people got plastic surgeries and stuff. You can't really tell what is hereditary anymore. Elements in life, right? So this is sort of the very fundamental thing that you guys have to know. So elements in life, we primarily separate these different elements into 4 categories or 4 tiers. We got the first tier, second tier, third tier, fourth tier. First tier elements, these are the things that are found, okay, most abundant in all organisms. Okay, so essentially all living things will have these 4 and in large quantities. And what are they? Carbon, hydrogen, nitrogen, oxygen. I mean, duh, it's organics, right? Second tier, the definition is they're much less abundant, but still found in all organisms, okay? But a lot less abundant. What are those? Things like calcium, chlorine, magnesium, phosphorus, potassium, sodium, and sulfur, okay? Of course, I highlight potassium and sulfur. Why? Phosphorus and sulfur. Phosphorus is found in DNA, okay? Nucleic acids. DNA, RNA, you got that 5' phosphate, okay? So this guy is in DNA, okay? Sulfur, amino acids, right? So this guy is in amino acids. In particular, 2 amino acids called cysteine and methionine. Okay, we'll talk about those when we get there. Third tier definition is they are metals present in small amounts in all organisms and essential to life. Okay, so these are also found in pretty much all organisms, okay? And they're metals, they're metal ions. They're essential to life because they assist in catalysis, okay? They help catalyze reactions, right? So in chemistry, general chemistry, you guys learned about catalysis, right? And sometimes catalysis, you would add transition metals, and then it helps to catalyze some reaction. Fourth tier, these things are found in or required by some organisms in trace amount. Trace amount is essentially very, 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 very little amount. You only need very little amounts of those, okay? What are they? They're like aluminum, arsenic, boron, bromine, chromium, fluorine. Yeah, so you don't see these a lot, okay? You obviously don't see these a lot, okay? So they are used by some organisms out there, okay? Just so you know they exist. These are the elements in life. Today is really just a quick review, right? So let's first take a look at some of the bonding abilities of carbon, okay? So why are we talking about carbon? Because carbon is very amazing. So those of you that took chemistry, you know carbon can form 4 bonds, okay? So like we talked about on the previous slide, the first tier elements, we got the carbon, hydrogen, nitrogen, and oxygen. And then you look at the bond forming ability of these molecules in terms of forming covalent bonds. Hydrogen can only form 1 bond. Hopefully that's not a surprise to you. Please don't draw 2 bonds on hydrogen, please, okay? Oxygen can form 1 or 2 bonds, okay? Nitrogen can do 1, 2, or 3 bonds, and carbon can do 1, 2, 3, or 4. If you don't know where these numbers are coming from, please let me know. I can tell you, right? But this is something that you should know, okay? Now, so some of the common bonds found in biology are shown in the right, okay? So these are the bonds that you definitely should know and their characteristics, okay? We got our normal CH bond, okay, CO bond, C double O bond, C nitrogen bond, C double nitrogen bond, and carbon-carbon bond, carbon-carbon double bond. Okay, so these are the common bonding schemes that you have in biochemistry. Okay, so these are the most frequently observed types of bonding between elements in biochemistry. So you will not have those crazy reactions that you find in organic chemistry. So actually, in organic chemistry, you have to memorize like at least 30 reactions per exam. Okay? Yeah, biochem, it's not like that. Biochem 2, you have to memorize a lot of reactions, but biochem 1, definitely no reactions. Right? So for our discussion here, there's not really reaction-based knowledge. Okay? If your carbon has 4 bonds, this molecule is tetrahedral. Okay, so that's important because tetrahedral, you have 4 things, 1, 2, 3, and 4. If the 4 substituents are all different, then this molecule is called. If you bind 2 carbons together, you have a CC single bond, a sigma bonding right here. And then you got, the other stuff are just away from this bond. Then the sigma bond is free to rotate. Okay, that, of course, you guys should know. If you have a sigma bond plus a pi bond, right, so you have a double bond here, then you restrict your molecule to somewhat of a plane, okay? But, that's just how it is, okay? All right, so then it does not have the rotation feature. Common functional groups in biochemistry, right, so these things are the functional groups that you should definitely know. You got the carbon functional groups, things like a methyl group, ethyl group, phenyl group, and of course you have methyl, ethyl, what's the thing comes after ethyl? Propyl. After propyl, okay, is butyl. Butyl, whatever, right? Butyl, and then you have pentyl, whatever, hexyl, whatever, okay? Right, so those are talking about essentially saturated, well, not necessarily saturated, but just carbon functional groups. You have oxygen-containing functional groups, things like carbonyl, okay? And carbonyl, you have a terminal carbonyl, okay? So this is a terminal. Carbonyl, right? So this carbon is at the end of a carbon chain. This thing is called an aldehyde, right? If the carbonyl is in the middle, okay, then it's called a ketone. Okay, that's that. If you go from carbonyl, right, so this is a hydrogen right here. If you do a oxidation, okay, an oxidation reaction, you can get carboxylic acid or carboxyl group. Basically, in biochemistry, every time we say blah, 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 acid, for example, what's an acid that you know? Acetic acid. That's not really necessarily related to biology, but acetic acid it's a carboxylic acid, right? Citric acid, 所以像你有时候去买饮料 ，on the back 不是会有什么 blah 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 酸吗？什么柠檬酸，什么乱七八糟酸，不同酸，对不对？ Usually carboxylic acid. Okay, so organic acids typically is talking about the molecule having a carboxylic acid group. Okay, and then you got hydroxyl group. Okay, hydroxyl group also a lot of times is referred to as an alcohol group because essentially this is an alcohol. Okay, so that's OH. Ether, sometimes you do find ether, that is if you would react 2 alcohols, do a water elimination, then you will get an ether. Okay, so it has an oxygen in the middle. Or you take a carboxylic acid plus an alcohol, you get an ester. Okay, so ester is where this is the carboxylic acid and this is the alcohol. In your organic chemistry lab, have you guys done this lab before where you take different carboxylic acids, match it with different alcohols, and you reflux, and you get a bunch of esters? 你们有机实验有没有做过？有没有做过这个实验？就是你拿不同的有机酸啊，就 carboxylic acid，OK， 比如说2个碳的、3个碳的、4个碳的，然后加上 alcohol，OK， 随便2个碳的、3个碳的、4个碳的 alcohol， 然后你反正加在一起嘛，对，加在一起，然后你就加热做 reflux，OK， 然后你就会合成 esters. Right? Esters 有什么特性？有没有知道？香气。Okay, so esters smell like fruits. Okay, so when I was in college, my organic lab, we had a lab that did ester synthesis. Right, so we got like 20 people in the lab, and every person has a different acid plus a different alcohol. Okay, and everyone's ester will smell differently. Okay, so I remember mine was like banana, and then the person next to me got orange, and then there are a bunch of things that smell like orange and banana together. You also got like, yeah, so you get really fruity smells. Some of the esters, you cannot differentiate what kind of fruit it belongs to, but it just smells fruity. Okay? Yeah, but don't do that, because I remember very clearly after 2 or 3 hours of staying in that lab, I got a super bad headache because the entire lab is filled with these crazy esters. Okay? So don't do that at home, right? And sometimes you also get anhydrides. Okay? So anhydrides are essentially, you got 2 carboxylic acids reacting together, eliminating a water. That's what you have, acid anhydride. Okay? So these are the functional groups that contain carbon and then oxygen. Okay? Now, for biochemistry, which of these are super important that you absolutely have to know? For this semester, carboxyl, definitely. Alcohol, you have to know because it's on proteins or it's on amino acids. And then esters, not so important. Ethers, maybe not so important. Ketone, you should know. Aldehyde, you kind of have to know. Okay, so those things you definitely have to know. Nitrogen functional groups. So these are the molecules, or at least functional groups that contain nitrogen. You have amino. Amino is usually we write it NH2. Okay, that's what this is. Okay. You have amido. Essentially, it's a carboxylic acid reacting with NH3, and then that's what you get. You have guanidinol. Okay, imidazole. You have to know. Okay, this one you have to know. This belongs in a amino acid called histidine. Okay? So those of you that are doing lab work right now, histidine, right? Histidine has imidazole ring on it. So in the procedure in which you use to purify that protein, okay, the very, very last step, you elute your protein with imidazole, okay, with a higher concentration of imidazole. Okay, that is because that's the same functional group that histidine has. Sulfur functional groups. You have sulfhydryl, or a lot of times, sometimes I don't use sulfhydryl so much. I usually just call it thiol. Okay, just like alcohol group, thiol. This one definitely have to know because it belongs to an amino acid called cysteine. Disulfide, this one you should know. Okay, you have to know this as well, because this is super important for a lot of human proteins. For example, your antibody. That's my ugly version of antibody. I don't even know if I drew it right. Okay? But because, these antibodies got like these bonds connecting to them together, those are the disulfide bonds. Okay? They are essentially 2 cysteines coming together. You do an oxidation, you get disulfide. Okay? It's pretty much a permanent link, permanent covalent link that link 2 molecules together. And then you have thioester. Okay? Thioester, like the name, it's an ester. It's just that here you have a sulfur instead of an oxygen. So these are the naming of the things that we're going to talk about. Make sure you know the nomenclature, or at least the names of these things. Throughout our future classes, a lot of times I will refer to molecules and functional groups as, for example, carboxylate group, alcohol group, thiol group. You need to know what I'm talking about. Okay? So this you have to know. Right? So that's something very important. Lastly, you have phosphor-containing functional groups. For example, phosphoryl. okay, or phosphoanhydride, or mixed anhydride. So phosphorylate is essentially you turn an alcohol into phosphate. So this guy is a phosphate, and then you put it on to this oxygen, and then you get a phosphoryl group. Phosphoanhydride is essentially two phosphoryl coming together, and you form phosphoanhydride. Simple, through a water eliminating reaction. Also, you got this thing called the mixed anhydride. This is essentially you take carboxylic acid plus a phosphate, and then you get mixed anhydride. Okay? These things are typically called, it's actually written down here, acyl phosphate. Okay? Acyl is referring to this guy. Phosphate is referring to this guy. Acyl phosphate. Okay? So this thing is called an acyl group. Acyl group. Okay? So anytime you have R blah 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 attached to something, this thing is the acyl group. Okay, that's the acyl group. It's not a carboxylate group because it does not have the O minus on that side. Right? It has everything else on that side. Right? So that's an acyl group. Most biomolecules in biology, okay, they actually have multiple functional groups within them. For example, acetyl-CoA. Okay, acetyl-CoA will become your best friend next semester. Right? For now, you can just sort of okay, but acetyl-CoA is like everywhere. Okay. Okay, I'll just tell you right now. So if you like to drink, okay, and you drink alcohol, and you guys know the alcohol, really, what kind of molecule is it? Okay, ethanol is the alcohol that you drink. Okay, it's called ethanol. Ethanol has 2 carbons. All right, so turns out when you go out and drinking, you drink ethanol. Okay, so ethanol actually looks like that. When it goes into your body, okay, when you metabolize ethanol, it actually becomes this whole thing. Don't ask me how you get all those carbons, right? Actually, carbon 1, carbon 2 is right here. Carbon 1, carbon 2. In the process, this whole thing is called the coenzyme A, CoA. Okay? It's going to get attached to this molecule, and also you are essentially oxidizing it such that it spits out 4 electrons. This is not important. All right? I'm just showing you this process. You'll get there when we get there. Right? So when you go out and drink, and essentially your ethanol, this is how it gets metabolized. Right? So it's going to oxidize this number 2 carbon. In this oxidation process, okay, and you are oxidizing this number 2 carbon such that initially it has, for example, 1 oxygen bond, now it has 2 oxygen bond and 1 sulfur bond. So you actually lost 4 electrons. I will teach you how to count electrons if you're interested, but for now, you can just believe me, okay? Now, coenzyme A is a carrier for acyl group. It's an acyl carrier, okay? And then this molecule, later on, it's going to go into a process called TCA cycle, and then it becomes CO2. Okay? So this is the first step in alcohol metabolism. All right? Now, coenzyme A. So if you look at coenzyme A, you have a thioester, you have an amido, you got another amido, methyl groups right here, alcohol hydroxyl groups right there, phosphoanhydride, you got phosphoryl, you got imidazole, you got amino. Okay, so in one little molecule, you got pretty much all of the functional groups that we talked about in the previous slides. Okay, so just to show you, in biology, a lot of complex molecules have all of these functional groups at the same time. Coenzyme A is one of the more complex molecules. Typically, inside of your metabolites, molecules are not so complicated. This is a large molecule. Okay? All right. Chirality, a very brief review on chirality. Right? Again, biochemistry is extremely structure-oriented. Structure means everything. in biochemistry. Okay? Molecule with the same empirical formula may have different chirality. Okay? That is, they can be enantiomers, diastereomers, etc. If they are different chiral compounds, they are different compounds. Okay? For our purpose, chirality makes them different things. For example, this is aspartame. Okay? Aspartame is a type of sugar substitute. Sugar substitute 知道是什么吗？代糖. Okay, sugar substitute. Aspartame is a pretty common sugar substitute that you will see around in shops. Essentially, it has several chirality or chiral centers. For example, one here, right? Now, if we look at this particular chiral center, okay, and if it is L-phenylalanine, it is aspartame. Okay, it's sweet. It is D-phenylalanine, then it becomes bitter. Okay, then it's not a sugar substitute. Okay, so one form is sweet, one form is bitter. Okay, so why is that? Because your receptor on your tongue, okay, it's a protein. Protein is 3D. Okay, it has 3 dimensions, right? Three-dimensional structure. So in order for this molecule to bind to the receptor on your protein and tell your head saying, oh, okay, this thing is sweet, it has to bind properly. Okay? If it does not bind in the exact shape, okay, then it's going to signal a different thing. Okay, it's going to signal a different signal. For drug design and discovery, this is even more important. There are a lot of examples where one chiral molecule, okay, one enantiomer is a drug, and then the other enantiomer is a toxic poison. Okay? So chirality is super important. And why is it important for biology? It's exactly because our proteins are 3D. Okay? Your proteins have a shape that recognizes a specific shape of the molecule. And if they are different chirality, then they have a different shape. Okay? So that's important. Aspartame, if you look at the structure, or actually if you look at the word, the name, aspartyl phenylalanine methyl ester. Turns out it's made from 2 amino acids. 它用2个 amino acids 做出来的，一个叫 phenylalanine， 一个叫 aspartic acid, aspartate. And then you combine these 2 amino acids, okay, one is here, one is here, and then you add a methanol right here. It becomes aspartame. Okay, why is this important? Because there are some people that have a genetic disorder that cannot metabolize phenylalanine. Okay, so they cannot metabolize phenylalanine, and as a result, phenylalanine accumulates in their body and actually will cause severe mental retardation. If it's in a high doses, it will actually kill the individual. Okay, so this is why aspartame, even though it's commercialized, but it's not super good. Okay, because what if someone who has the disease doesn't really know, and then they eat this, then they will actually get into serious trouble. Okay? Biological macromolecules and their monomers. Okay, so many biomolecules are macromolecules. Okay? What are macromolecules? They're polymers of high molecular weight. So below is a chart of common macromolecules found in life. Okay? Macromolecules like protein, polysaccharide, nucleic acids, lipids. Polysaccharides, it just means poly and then, carbohydrates. Okay, so poly sugar. Their monomers for protein is obviously amino acid. The monomer for polysaccharides is monosaccharide, so this is sugar. The monomer for nucleic acids are called nucleotides. Monomer for lipids are fatty acids. And their linkage, okay, here's the thing. Proteins are linked via amide bond. Okay, but in biology, or we call it peptide bond. Peptide, amide, same thing. Okay, it's just that 2 different people saying the same thing. Okay, they just have to. Biologists or biochemists have to make themselves feel special. Okay, by giving it a different system. For example, here, L, right? Polysaccharides, they link together via a ether bond, or you can also call it a glycoside bond, glycosidic bond, and they form polysaccharides. Nucleotides, they use phosphodiester bond. Okay, so this is the phosphate. When we learn general biology, we learned that 5' phosphate can bind to the 3' OH. Lipids, they form via an ester bond. Okay, so lipids typically will look like this. Okay, they look like that. Where's the ester? This is the ester. That will be a triacylglyceride. So if you go to the hospital and you take a blood, and then the doctor said you are eating too good because you have a lot of triacylglyceride in your body, that's his way of saying, 就是减肥啦 ，OK， 就是你那个血脂太多 ，triacylglyceride， 这个东西就是 triacylglyceride. Common functional groups. Let's take a look at each of the individual chemicals and then what kind of functional groups are present in glucose or in carbohydrates. Essentially, if it's a sugar, it will have what group? How many groups do you see in this molecule? You see a lot of what? OH, right? So OH is a what kind of functional group? Alcohol, right? Alcohol or hydroxyl group. Okay, so already we have a bunch of these alcohol groups right here, right here, right here, right here. Okay, so these are all hydroxyl groups, okay, or alcohol. And then what is this? All right, this thing is an aldehyde. Any sugar or monosaccharide, you have these two types of groups. You have alcohol group and you have aldehyde groups. There are another type of sugar that moves the double bond into the middle. Then instead of aldehyde, it has a ketone. Okay, but we'll get there. So besides the adenine group, this is the adenine group. Okay, this is the adenine. What other groups are present in DAMP? You have alcohol, right? You have, what is this? This is actually hard. This, it's actually coming from this carbon, okay? And it's actually supposed to be an aldehyde group, but we'll get there when we get there, okay? It secularizes, okay? It made into a ring so that you form an additional bond from another place, okay? This, phosphoryl group, right? What about DATP? Okay, DATP is the real building block of DNA. Okay, that means that when you actually make DNA polymerization, you're actually using DATP instead of AMP. Okay, instead of this guy, you're actually using this molecule. Okay, and what do you have? Okay, you have the same hydroxyl. Now, instead of a phosphoryl, you have a phosphol anhydride, right? So those are phosphoanhydride bonds. Yeah. If I don't look at this, they're the same molecule, right? So this molecule, usually when we say the monomer for DNA, it's going to look like this. So this guy is going to be attached to another OH of the previous base. Okay, so DNA is always, this attaches to this. Okay, so this OH attaches to the phosphate of the next group. This thing does not have enough energy to react. Okay, the energy for this molecule is too low. So you have to use this. This is the true substrate for polymerizing DNA. Okay, and why is the true monomer? It's because phosphoanhydride has a lot of energy inside. Okay, so when you break these two away, it has a negative delta G. Okay, so it makes the reaction go faster. I'm actually not faster, but it makes the reaction able to occur. Peptide bond is an amide bond between two amino acids. Okay, so what is a peptide bond? This thing is a peptide bond, the thing that's circled in red. For example, the thing in colored, we usually call it a residue. And this particular residue is a tyrosine residue. This thing is called tyrosine. And tyrosine is one of the 20 kinds of monomer that makes up your polypeptides. If we look at the tyrosine structure, in addition to the phenol, okay, the phenol functionality, this is a phenol. In addition to that, what other functional groups are present? This guy is a carboxylate, okay, or a carboxyl group. Okay? Carboxylate, it's a carboxylic acid, so it's an acid. And then, what's another group? Okay. One very easy way to spot functional groups is you look at the elements. Okay. If you have an oxygen, that's going to be a functional group. If you have nitrogen, that's going to be a functional group. Okay. So you got nitrogen over there. Okay. What group is that? You might be confused because there's a charge on here. Okay. It turns out this thing is just a amino group. Okay? It's just an amino group. It's a proton-mated amino group. Why? Because it has one extra proton. Okay? So that the nitrogen becomes positively charged. Okay? So, but it's an amino group. Okay? As a result, you have this, let's say this guy is carboxylic acid. That's an amino group. Together, they give you amino acid. Okay? Together, they give you your amino acid. Okay? Amino is on the nitrogen, acid is on the organic acid. Okay, so that's why it's called amino acid. So resonance structure is a way to describe the delocalization of electrons within a molecule. So we will spend a little bit of time talking about mechanisms, like reaction mechanisms, meaning like nasty arrows flying around. People hate that, I know. This is a chemistry class, okay? So electron movements, okay? We have what? We have two types. We have two electron movement shown by an arrow that looks like that, or we have a one electron movement shown by a nasty half arrow that looks like that. Okay? Half arrows don't happen that much in biochemistry, but they do happen a lot. Not that much compared to the double arrow, but they do happen. Like for example, let's take a look at acetate. You have a negative charge right here, and you're going to move, so this is a full arrow, so you're going to move two electrons at once. Okay? Two electrons form a formal bond. 听懂哈？两个 electron 给你一个 bond 哦，一个 bond 上面是两个 electron 哦。Okay? That you have to know, right? So you form a bond right here, so that this guy becomes two bonds, and then now this carbon, okay, would have too many bonds, so one of them pop up, so that you share, you transfer the negative charge from this oxygen to this oxygen. Okay, this thing is called resonance. Okay, now resonance is a very important concept because any molecule that has resonance structures, okay, it becomes stable. Having resonance means stability. Okay, why is that? Because it essentially allows the electrons on your atoms be shared across the molecule. Okay, so you can think of it as， 假如说你今天在一个房间里面哦，有一个人在角落放屁，有没有？然后那个会散嘛，对，会散，就是 thermodynamics 会告诉你这个东西要散开来 ，OK， 它要扩散，扩散以后，你会达到 equilibrium，right？ Electron working same way， OK， 就是如果你今天把 charge，if you put the charge，OK，your electron densities，OK，your electrons are negative charges，OK， so if you put all your negative charge on one atom, it is definitely going to be less stable than if you put it on two charges. Okay, so you share it out. Okay, so something very painful, you share with your friend. It's that kind of concept. Okay, so resonance will make your molecule stable. Okay, because it lowers the energy. Why is this important? Because a lot of times when we're talking about reactions, okay, if the product of your reaction has resonance, then this reaction is a very favorable reaction. 听懂我意思吗? Maybe okay? Okay? So if the product has resonance, then the reaction becomes favorable. Okay? If the substrate has resonance, then the reaction is not favorable because the substrate energy is too low. Okay? So resonance becomes super important when we talk about chemical reactions, okay? Biochemical reactions. Right? So benzene, it has a lot of resonance because the electrons on each of the carbon essentially is shared with each other. Okay, so this entire molecule, okay, has the electron shared. Okay, so not one molecule has more electron than another. Okay, so everybody has same amount of electrons. So that makes benzene a super stable chemical. Okay, so it's difficult to react with benzyl or anything aromatics. Okay, so remember in organic chemistry we always say, you know, benzene type of stuff, you know, aromatic residues usually is difficult to react with it. That is because it's very stable. Okay, it itself does not have a lot of energy to do some other things. Okay, reactivity and the stability. Okay, so generally speaking, the more stable a chemical is, the less reactive it is. Okay, now, so essentially I want you guys to develop a feeling of, if you take a look at a molecule, a chemical structure, and try to get a feeling of, is it stable? Okay, based on what? Based on the functional groups. Okay, so do you think this molecule is stable? If it is, then probably it does not react very well. Okay, do you think this molecule is stable? If it's not stable, then it reacts very well. Okay, so if a compound undergoes a reaction that can generate a more stable product, then that compound is more reactive than its product. Okay, for example, let's take a look at ATP. Okay, we all know what ATP is, hopefully, those of you that took biology. It's the cellular energy, right? ATP is cellular energy carrier used to drive chemical reactions. All right, it has these phosphoanhydride bonds. Okay, and I can tell you right now, those have high energy. Show you why. Okay, one of the reasons why phosphoanhydride bonds are not very stable is because it has a lot of negative charges. And according to, you know, Coulombic interaction, a lot of negative charges next to each other is a bad idea, right? Because negative repulse, so this molecule itself has a lot of energy inside. Make sense? We're good. Okay? So if we hydrolyze it, okay, for example, we use water to react with this particular phosphor, okay, right here, so that you generate one phosphate, okay, and then a phosphoanhydride bond, one phosphoanhydride bond less molecule called, this is ADP, diphosphate. Okay, T is triphosphate. Okay, now this delta G is pretty large. Okay, the delta G prime naught, we'll talk about the definition of that prime in just a minute, but you should know what delta G naught is, the little circle. Okay, it's minus 30.5. Okay, it's quite large. Okay, this is a pretty good amount of energy. Pretty good. Okay, now the reason why I can give off that amount of energy is because this guy has resonance. Oops. Okay, you can pop this electron down, this electron down, this electron down, so the negative charge can spread. Okay, so you got resonance here. Here, you have less charge repulsion. Okay, so overall, these guys more stable. Sounds good? Sounds good, okay? Because it's more stable that way. Okay, huh? So throughout biochemistry, this semester and next semester, I want you guys to develop a sense in terms of take a look at a molecule and take a guess whether or not it's stable or not stable. So that's sort of the skill that I want you to have. This thing, probably not very stable because of phosphohydride bonds and because they have a lot of negative charges, and that will cause it to repel each other, and then if you hydrolyze it away, then you actually get a more stable product. Okay? Now, so this is review again of free energy and the direction of chemical equilibrium. Okay? So always when we represent a chemical reaction, we say delta G is negative or positive, right? Delta G is always negative for biological reactions. Let me write this down here. This is actually important. Now, notice I don't put the prime, okay? Delta G prime standard may not be negative. This is always the fact, okay? What's the difference? What's delta G and delta G not, right? Standard, this guy means standard. So it assumes that everything is at 1 molar, okay? But of course, in reality, our things don't have to be at 1 molar, right? Who says the chemicals in your body has to be 1 molar? Doesn't have to be, okay? So the real delta G is always negative, okay? Because if it's not negative, it cannot occur, right? So it's actually a logical thinking. If it's not negative, it will not happen. If it will not happen, doesn't make sense, right? Does that make sense? So everything that's happening to you right now, the delta G is negative. 合理哈? Okay? Now, so here, let's take a look at why we want to study delta G. Okay? So for any reaction, you have A plus B gives you C and D. Okay? It can go to an equilibrium if you let the time extend. Okay? So turns out delta G is absolutely related to the equilibrium constant. Okay? So that if, let's say, your delta G is minus 17.1, the equilibrium is 10 to the third. Okay? And equilibrium constant is usually defined by product over reactants. 听懂吗？就是你除嘛，上面是 product 浓度，下面是 reactant 的浓度。Okay? So basically it's telling you if delta G is negative, then the reaction favors the product. Okay? Because KEQ is a large number. Okay? So you want to make product. If the delta G is a positive value, you want to make the reactant. Simple as that. Okay? So this general thing you should know. Okay? So when KEQ is greater than 1, delta G is negative. When KEQ is less than 1, delta G is positive. Okay? Does that make sense? We're good? Now, what I just wrote here, delta G, okay, it's not only just the standard, but it also accounts for the concentration and the temperature of the environment. Okay? So depending on the concentration of the metabolites inside of your body, the real delta G, so this thing guy, well, I always call it the real delta G. And then this thing is the standard delta G. Okay? All right? So real delta G in your body, always negative. Okay? Always. But standard delta G doesn't have to be. Okay? There are a lot of reactions, standard delta G is very positive, but somehow inside of your body, it becomes negative for the real delta G. Okay? How do you do that? By messing around with the concentrations. Okay? And also acid dissociation constant. The same idea applies to acid dissociation constants. Okay, so the protons moving from one molecule to water. Okay, that's acid dissociation constant. Okay, that's what we call Ka. Okay, Ka is essentially you're looking at the equilibrium constant of acid plus water gives you the deprotonated, this is the deprotonated acid, or we usually call it the conjugate base. Okay? And then the proton is right here on water. Okay? So essentially it tells you, okay, it tells you how stable the conjugate base is. Okay? If the conjugate base is very stable, okay, then your Ka will be very high. Make sense? Okay, makes sense. All right. And then of course pKa is the negative log of Ka. Okay, so lower the pKa, higher the Ka, blah blah blah. Okay. So now, how easy is the proton removed from the compound HA? That totally depends on how stable the conjugate base A minus is. Okay, so let me give you an example. What is this guy? Do you guys know what molecule this is? 2-carbon acid. 2-carbon acid is acetate. Okay, well actually this thing is acetic acid. 醋酸. Okay, acetic acid 或者乙酸. All right, acetic acid. Okay, conjugate base acetate. Okay, now acetate has resonance. Okay, so that makes this proton to be capable of passing it to water. Okay, so the pKa of this is going to be pretty low, okay, because Ka for this reaction is going to be high. Okay, so just 2 minutes, okay? So Ka for this reaction will be high, pK will be low. Why is the Ka high? Because the conjugate base acetate is very stable. Because acetate is stable, okay, acetic acid is less stable. That makes the Ka high because Ka is defined by product over reactants. Okay, so Ka is high, pKa is low. Okay? Now, the lower the pKa, obviously the easier the proton is removed. Okay? So that's that.